Hi everyone and welcome to Chapter 3, which we call the chemistry of organic molecules, basically because students are terrified when we call it what it is, which is organic chemistry, but I promise you it's not that bad. So the first thing you have to figure out when you're studying organic chemistry is, well, what exactly does organic mean? And when we talk about organic molecules, we are referring to anything that contains both carbon and hydrogen. Okay, so it's got to contain carbon and hydrogen in order to be organic, which you'll find in most living things. Now, there are 4 classes of these organic molecules, which basically compose life. They're called the biomolecules because you need them for living things. And they're listed out here: carbohydrates, lipids, proteins, and nucleic acids. And throughout this lecture, we're going to basically go through what each one represents, what its function is, and what some examples are, even though I'm sure that you guys have heard all of these terms at least one time or another. Now, just like we just said in terms of organic chemistry being, the study of things containing carbon and hydrogen, which are living things, inorganic chemistry is then the opposite of that. Inorganic chemistry is the study of the non-living world, which are the molecules that do not contain both carbon and hydrogen. Okay, so for organic, think of living and carbon and hydrogen, and inorganic, think of non-living. Now, a lot of this we just mentioned is determined based on carbon content, and carbon is one of the important elements. Its atomic number is 6, which you guys by now, thanks to the previous lesson, are very comfortable saying what that means. So whenever you see atomic number of 6, that tells you that it has 6 protons. Now, with those 6 protons, if it's a neutral atom, it also has 6 electrons. And those 6 electrons, when you think of how we arrange atoms, remember the first innermost orbital will have a maximum of 2 electrons, which then leaves us with 4 valence electrons, meaning 4 outer shell electrons. And as we mentioned in the previous lesson, those electrons, those valence electrons, determine how an atom interacts with all other atoms. And with carbon, by having 4 valence electrons, it then wants 4 others, right? Because if you think about it, it wants to make it up to 8. Now, what's cool about that is that means that it is perfect at linking up with other carbons. So as you see here, you end up with these long chains of carbons, and to fill in any extra missing electrons, they use hydrogen, because hydrogen, if you remember from the periodic picture that we had for the periodic table in the previous lesson, hydrogen needs 1 electron. So if you picture carbon, it'll always want to have 4 single bonds around it, or you'll have double bonds included. 4 single bonds because it has 4 electrons that it wants to share. Okay? Now, when you have organic molecules, you don't just have to have carbon and hydrogen. They end up having a lot of other atoms that they interact with. And these atoms, a lot of times you'll see they make these different combinations into what we call functional groups. And functional groups in molecules are very important because the functional groups are these specific combinations of atoms that bond to those chains of carbon that you just saw in the previous slide. And these functional groups are significant because they give molecules their specific properties. Okay, so when we ask why are functional groups significant, it's because they give molecules their specific properties, meaning they will determine how molecules interact with other molecules, how molecules bend and fold, where these molecules will want to be in cells or in the environment. And the reason that is that there are different types of functional groups. Some, as you see listed out here, will be hydrophilic or hydrophobic. And as you already know, hydrophilic are polar parts of a molecule, and hydrophilic, hydro and philic, think water-loving, so hydrophilic functional groups will want to be near water, whereas hydrophobic ones, which are nonpolar, okay, we're going to keep bringing back things from previous lessons, hydrophobic are nonpolar, and they're going to want to be positioned or located far away from water. So which functional groups are hydrophilic versus which are hydrophobic will really help determine where that part of the molecule will want to be. Will it want to be near water or will it want to be away from water? You also have ones that are acidic, neutral, or basic, and that will also affect their charge. And if you think about it, that will affect the pH around these molecules. It will affect how they interact with them. And so, for instance, if you have a functional group that has a positive charge, it's going to want to be near a functional group that has negative charges, right? So it's going to start sticking to any negatives that it finds. So the functional groups are very important because they determine how molecules will interact with each other and with their environment. Another term you see a lot in organic chemistry is the term isomers. And the pictures that you have on this slide are examples of two isomers. Both of these molecules will have the same molecular formula, meaning that they both have the same number of carbons. So notice 1, 2, 3 carbons, 1, 2, 3 carbons. They'll have the same number of hydrogens, which is 6 in each, right? 1, 2, 3, 4, 5, 6 hydrogens, and then you have another 6 hydrogens here. And 3 oxygen in both. See the 3 oxygen in both. So they both have the same molecular formula, meaning the same number of each of their elements. But what's different is how they are arranged. So an isomer is defined as two molecules with the same molecular formula, but a different arrangement of the atoms, okay? Different arrangement of the atoms. Make sure you write that down. Now, what I mean by arrangement of the atoms, you notice that the double bond to oxygen is on the last carbon in this molecule, but it's on the central carbon in this molecule. So they have the same number of atoms, but where their bonds are located, the order or arrangement of their atoms is different. So now when we talk about organic chemistry, the foundation are the 4 biomolecules. And when we mention these 4 biomolecules, which you see here, we've mentioned them earlier, you have your nucleic acids, carbs, lipids, proteins. When we mention those, we call them macromolecules. And macro, as you know, is the opposite of micro. So 4 biomolecule macromolecules means that they are large molecules and they usually have many repeating subunits, as you can see here. Each of these has their repeating monomers building them up. So monomers are small subunits and polymers are big macromolecules. Now, these guys are synthesized and broken apart just like we mentioned in the previous lectures, which is through water, either removing water or adding a water molecule, so H2O. And you see that down here. So biomolecules, which are big macromolecules, are put together by dehydration synthesis, meaning removing that OH and H to make a bond. So each monomer is stuck together and you end up having a big polymer. Okay? Polymer meaning many subunits. So that's dehydration synthesis, removing a water molecule to synthesize the macromolecules, okay? So make sure you remember it both as dehydration synthesis and also removal of water, because I could ask it either way. Whereas the opposite of that, breaking apart a molecule, is done by hydrolysis, so adding water. And again, I remember I said you can picture taking a big fire truck hose and spraying the water with such force that it breaks apart these big molecules. Now we're going to go through each of the biomolecules one by one, just so that you can get an idea of their function and some examples of them. So when we talk about carbohydrates, they are basically made up of saccharides, right? Sugars. So there's monosaccharides, disaccharides, polysaccharides, and just like the name suggests, it's either one ring of sugar, two rings of sugar, or many rings of sugar. Now, when you ask yourself what's the main function of carbs, think of your everyday life, right? Think of carbs as in the example of sugars. Well, what does it do? It gives you quick energy, right? So the main function of carbohydrates is quick energy. The second function that carbohydrates can have is structural. So they can be structural material. And I want you to know each of these examples, because I can ask you which of the following is an example of a carbohydrate, or I can ask you cellulose is an example of which biomolecule, right? So with these three examples, you've encountered them in everyday life, but you may not have known it. So cellulose is the carbohydrate that you find in plants. So for instance, this giant cactus is nice and rigid and solid thanks to cellulose. And fun fact, you wouldn't be able to digest plants and vegetables because of cellulose, but thankfully you have bacteria in your gut that helps you digest them with their enzymes. So cellulose is a nice, tough, structural carb in plants. Chitin is the tough, structural carbohydrate in shells. So a crab shell, a lobster shell, that crunchy outer shell is thanks to chitin, which is a carbohydrate. And then the last one I want you to remember is peptidoglycan. And peptidoglycan is the carbohydrate that gives bacteria their cell wall structure. Okay, so when you think of cellulose, think of plant carbohydrate. When you think of chitin, think of crab or crustacean, shell carbohydrate. And when you think of peptidoglycan, that's the bacterial carbohydrate. And all three are to have solid, strong structure. Okay, so carbs are for quick energy or for structure. As I mentioned in the previous slide, they can be mono, meaning one ring, disaccharide, meaning two rings or two subunits, and polysaccharide, meaning many. Okay, so you get to see that here, one, two, many subunits, which sometimes you hear called monomers, like you saw in the previous slides. Now, when you think of monosaccharides, the examples that you should know are glucose, galactose, and fructose. And where you find these, glucose, you can think of your blood sugar, right? Fructose is the sugar in fruits. And each of these is a monosaccharide, meaning a single ring. The disaccharides include lactose as the big example, and lactose is the sugar that you find in milk. And the other big example is sucrose, which you use every day. Regular old sucrose is the table sugar that you eat. And then polysaccharides, those are the ones that are more structural. Those are the ones that are a storage form of sugars or of carbohydrates. And the examples that you have of the polysaccharides are big starch and glycogen as well. And what the starch is the stored form of carbohydrates. You especially find that in plants, just like when we mentioned cellulose, whereas glycogen is the stored form of carbohydrates that you usually find in animals, including yourself. Both of these, starch and glycogen, so circle, star, highlight those examples, both of these are made up of many chains or side branches of carbohydrate rings, so sugar rings, you can think of them as. Okay, so you have monosaccharides, disaccharides, and polysaccharides. The next biomolecule that you have is lipids, and the way students usually remember these are as fats, but there are actually 5 main types of lipids. All of them are made from hydrocarbon chains, so that's what you see represented right here. Hydrocarbon meaning carbons and all those hydrogens in long chains. And that structure ends up making them insoluble. And what does that tell you about lipids? That tells you if it's insoluble, that means it does not dissolve in water. And that's why you'll see if you get oil in water, right? What do you notice? It floats to the top. So you see those separate circles of oil because it's not able to dissolve in there. That's also why if you have really fatty food or really fatty soup, for instance, and let's say you put it in the fridge, you then see that big chunk layer of fat right on the top because the fat does not dissolve in it. Now there are 5 main types of lipids, and we have them listed here. Make sure you know the examples of what is a lipid. So the types or the examples are fats, oils, phospholipids, steroids, and waxes. And each one you'll find in different locations. I'll give you a little info about each of them in the next slides. So first off, when we start talking about fats in general, fats, the big main category of lipids. Sometimes you also see them called triglycerides. And the big function of fats, even though we all hate fat, right? We try and get rid of it. We're so quick to say we don't want fat, but we actually need fat. Fat provides insulation and it provides long-term energy storage. Okay, so remember that carbohydrates were for quick energy, whereas fats are for long-term energy storage. So make sure you write down the 2 main functions of fats are insulation, which you can see, big creatures like this guy here, they have a lot of fat for insulation, and it's called blubber. And fats are also important for long-term energy storage, where animals can break down that fat when they end up needing it in a time of crisis. Now, fats are made up of 2 subunits. These subunits are the fatty acids and glycerol. So the fatty acids are the long chains of hydrocarbons, meaning carbons and hydrogens. These are the fatty acid chains. This is one big fatty acid chain here. Okay, this is a big fatty acid chain, but it's been bent because it has a double bond in it. And then glycerol is right here, the carbon with the oxygen and the OH group attached. Okay, so this is glycerol and fatty acid. Glycerol and fatty acid, those are the 2 subunits. Now, fatty acids can be categorized 2 different ways, which I'm sure you've heard in everyday life and nutrition. There are saturated fatty acids and they're unsaturated. And as you can see in the picture, the way that they differ is saturated fatty acids. They are saturated, meaning they are completely full of hydrogens, right? They are all single bonds. Whereas unsaturated fatty acids, they're not saturated because they have at least one double bond, which can make them interact with other molecules because that can be broken open, okay? And it ends up with a boomerang shape. Now, the main reason why saturated fatty acids are worse than unsaturated, when you look at this difference in shape, the boomerang versus the flat, saturated fatty acids in this flat stick-like form, they can easily stack on top of each other, kind of like picturing 2x4s out in the backyard. So saturated fatty acids, you could get a whole bunch of them kind of lined up, stuck together in your body, in your arteries. Whereas unsaturated fats, because of that boomerang shape, picture trying to stack those, sometimes it's a little harder because they take up more space. Okay? Another type of lipid are the phospholipids, and the place that you find phospholipids is in plasma membranes. So every single one of your cells will have this phospholipid membrane all around it for structure, for transport, for things that we're going to go over in the next lecture. Now, with phospholipids, just like any other lipid, they are made up of fatty acids and glycerol. So remember that from the previous slide. Fatty acids and glycerol tells you that you have a lipid. Okay, so that could easily be a multiple choice question where I say, you know, if you encounter fatty acid and glycerol in a biomolecule, which biomolecule is that? It's a lipid. But in addition to that, phospholipids will then have a phosphate group chilling on the end of that fatty acid and glycerol structure. And that's very important because you now have this dichotomy where you have a polar phosphate group on the end of the nonpolar fatty acids. Now that's going to be very important because what do you know about polar versus nonpolar? Polar, just like water, like dissolves like. So polar phosphate groups will be hydrophilic. They'll want to be near water. Whereas fatty acids that are nonpolar, what are they? They're hydrophobic. They're scared of the water. They don't like the water. They want to be far away from it, right? Hidden away. So that's why you get these plasma membranes that are this double layer, this phospholipid bilayer, which you see here, where all of the polar phosphate groups are on the outsides that are exposed, touching water. All of those non-polar fatty acids are here in the center, hidden away, trying to protect themselves and avoiding water. So this is very important for cell structure, and we'll talk about that in later lectures. Again, phospholipids, what's very important is that idea of having polar and non-polar on one molecule. Lipids also include steroids and waxes, and usually when you hear steroids, you think of the drugs. But what we focus on in terms of steroids is the fact that when you see steroid structure, like here and like over here, steroids, notice, they look very different from the other lipids that we've seen. Steroids are 4 fused carbon rings. Okay, make sure you write that down. 4 fused together carbon rings. That's steroid structure, as opposed to the other lipids that have what structure? The fatty acid and glycerol, right? Those long chains that you saw. So steroids are kind of unique among the lipids. There are some important examples of steroids that you've encountered in your everyday life. There's cholesterol is a steroid, meaning cholesterol is a lipid. That's part of animal cells' plasma membranes, and it's also a precursor of your other steroids, meaning that cholesterol then gets broken down to basically form some of your other steroids. The other 2 steroids that I want you to know, because most people do not think of these when they hear steroids, testosterone and estrogen. Both of those sex hormones are examples of steroids. Okay, so when you see what are the examples of steroids, I want you to remember cholesterol, testosterone, and estrogen. Now, waxes are also a little different from some of the other lipids. They still have long-chain fatty acids, but they're also attached to long-chain alcohol. And that's important because it helps give them a nice high melting point. It makes them waterproof, which is why you see, you know, that glossiness to waxes and why you see water just bubble up, bead up into droplets on top of it. And they're very tough to break down. Okay, so when you think of waxes, think of fatty acids plus alcohol, and think of very resistant type of structure. That then brings us to the third type of biomolecule, and I call this guy the magician. Okay, proteins are the magicians in your body because they basically can do just about anything for you. So when we talked about carbohydrates, they had two main functions. It was quick energy and structure. Lipids had insulation and long-term storage as their functions. Proteins, look at all these different functions. That's not even all of them. Okay, so proteins are involved in metabolism, support, transport, defense, regulation, and motion. Okay, so there are tons of proteins in your body. We're going to talk about them more throughout the course, but as kind of an introduction, when you think of metabolism, I want you to circle star highlight enzymes. Okay, circle the fact that enzymes are a type of protein. That's very important because that means when we talk about enzymes, anything that can happen to proteins, such as denaturing, can happen to enzymes because they are proteins. Now remember, metabolism means all of the chemical reactions in your body. Enzymes help with that. They speed up these reactions. And again, we'll go into more detail later. Proteins are also involved in support. So when you hear keratin and collagen, especially in beauty products, those are both examples of proteins. Proteins involved in transport are the various channels that we'll talk about, and proteins that help move things from inside the cell to outside and in the opposite direction as well. Antibodies, so whenever you hear antibodies in vaccines, in getting better from sickness, those are proteins. And any of the hormones in your bodies, other than those steroid hormones that we just mentioned in the previous slide, but your other hormones will be proteins. And motion, any of your muscle movements. So when you hear actin and myosin and all that fun stuff, that's all proteins. Okay, but again, make sure you circle star highlight enzymes as a big example of protein. Now, this is another slide that I want you to put stars all around. What are the 4 levels of protein structure? Because proteins would not function without proper structure. And in your body, if a protein ever loses its 4 levels of protein structure, boom, you now have big time illness, and it can even be fatal. Now, the 4 levels of protein structure are as follows. The first one is the sequence of amino acid. So basically the string of amino acid names such as leucine, tryptophan, tyrosine, any of those in a row, the string of amino acids is the first level of protein structure. When that string of amino acids starts interacting with each other, meaning an amino acid over here starts interacting with one over here, you get the second level of structure, which is a single alpha helix or a single beta sheet, meaning one alpha helix or one beta sheet. That is a secondary structure. What happens is these strings of amino acids are really long. So rather than just having one of each of these, you don't just end up with secondary structure. These guys start interacting with each other. So when you have multiple alpha helices interacting with multiple beta sheets, this is now tertiary structure. They also call it a globular structure. Okay? Then when you have multiple of those globs, meaning multiple of those little proteins interacting together as multiple subunits, when you hear the word subunits, that tells you you're talking about the 4th level. So a lot of times that's where you'll see with enzymes and all, you're looking at the 4th level of structure. Okay, now I recap this on the next slide. So this slide here, you can have this recap so that you can jot down your notes. Again, primary structure is the sequence of amino acids, just the names written in a row, such as if I wrote tryptophan, leucine, tyrosine, all of that in a row. Secondary structure is if I tell you that you're observing a single alpha helix, or you're observing a single, just one, beta sheet. That's secondary structure. Think secondary single. Tertiary is when you're looking at multiple alpha helices, multiple beta sheets, and quaternary, you will see the word subunit. Multiple subunits, multiple polypeptides. When you see that, you know you're talking about quaternary. Put stars all over the slide because I like to ask about these different levels of structure. Now, like I mentioned before, the reason why I emphasize those levels of structure so much is that proteins cannot function if they lose their structure. And the two ways that proteins can lose their structure is the term denaturing or proteolysis. Sometimes you hear it as proteolysis. Denaturing means that a protein has unfolded. Proteolysis means it's been chopped up. Okay, so denaturing versus proteolysis. Denaturing is unfolded, and that means that it lost its secondary, tertiary, and quaternary structure, but not primary. Whereas proteolysis, when you see that P, it lost primary as well, because proteolysis means it's been chopped up. And again, anytime you hear lysis in biology, it means chopping up, breaking apart, right? So make sure you are comfortable with either of those terms, because both of them result in misfunction and possibly death. And I put some examples of illnesses that can happen when there's problems with protein folding. Now, when we talk about proteins, it's important to know that they are made up of what's called amino acids. And there are 20 amino acids. And what's significant about the amino acids is that they each have a unique R group. And the R group is their functional group, just like we talked about functional groups earlier. So when you think about these different functional groups, the significance is that the R group can have different properties. It can, for instance, be nonpolar versus polar, or it can be charged positively or charged negatively, or not charged at all. Now, what is significant about the R groups is that their different properties affects how that protein will then fold and how it will interact with other molecules. Okay, so the significance of R groups is they determine how that protein will fold and how it will interact with other molecules. Now, that's important to know that, for instance, polar versus nonpolar, how will that affect the folding? Well, you're all pros at this now, right? Polar wants to be near water because it's hydrophilic, and nonpolar would fold away from water because it's hydrophobic. Positive versus negative charged R groups. The positives will want to repel other positives and will want to attract other negatives, right? Because opposites attract. So that would affect folding. Positives would want to be near negatives. And lastly, there's a term I want you to circle, aromatic. Aromatic R groups means that the R group, which you see in blue, the R group has a benzene ring. Okay, so you see this big bulky ring. That affects folding and transport as well, because if a protein has a lot of aromatic R groups, well, it's not going to fold as well, and it might not be able to pass through certain channels, right? Because it'll be big and bulky. Okay, so make sure in terms of the significance of R groups, make sure you know that they affect the folding and interactions of the protein. Make sure you know how polar would differ from nonpolar, how positive versus negative charges would affect the protein, and how aromatic R groups affect the protein, okay? And again, for aromatic, think big and bulky. Now, these amino acids are joined by peptide bonds, and again, they will be bonded through dehydration synthesis as well. So forming those bonds, you will have dehydration synthesis, which you see in this picture here. Then we get to the last biomolecule, which is nucleic acids, and that is my personal favorite. I love anything to do with DNA and RNA. Genetics is fascinating. Now, with nucleic acids, there are a few things to know. First of all, their monomers, meaning their subunits, are nucleotides. So whenever you see the term nucleotide, that means the building block of nucleic acids. And those nucleotides, when we talk about them, they'll be made up of a few components. It's usually a sugar, base, and phosphate, which we'll go over in a later slide. Now, nucleic acids have very specific cell functions depending on which nucleic acid you're thinking of. Whenever you hear nucleic acids, I want you to think of DNA and RNA, and also later we'll mention ATP as well. So DNA is deoxyribonucleic acid, and that, as you know, is our genetic material. Okay, that's the blueprint for everything that you end up becoming, okay, in terms of cell structure. So what DNA does is it codes for RNA, right? And that's through transcription. Okay, so transcription, if you ever hear that term, that's DNA coding for making RNA. And again, so DNA is the genetic material, and then next you get RNA, and RNA is the code or the blueprint for making proteins. Okay, so that's involved in the assembly of proteins. And as you know, proteins then do a ton of things themselves as well. Now, when we talk about nucleic acids, like I said, they are made up of what are called nucleotides. And when you have a nucleotide, as you see in this adorable GIF, or as they now say you're supposed to pronounce it, GIF, you have a sugar, a phosphate group, and then a base. And when we say a base or nitrogen base, we mean these letters here. For DNA, it's A, T, G, and C, which stands for adenine, thymine, guanine, and cytosine. And then in RNA, they have the adenine, the guanine, and the cytosine, but instead of thymine, RNA will have uracil. Okay, so these four letters that you see are the bases for each. When you hear the term nucleoside, that's like a nucleotide, but it's missing the phosphate group. So nucleoside is just referring to the base and the sugar. You can underline the S in nucleoside to remind you that you're focusing on a sugar and a base, whereas nucleotide is the whole shebang. It's the nucleoside plus the phosphate group. So under nucleotide, if you want to write sugar, phosphate, and base, you can. You could put it in any order you want. Some people write sugar, base, phosphate, however you want it. Just know that a nucleotide has those three things: a sugar, a base, and a phosphate. Now those bases are held together by hydrogen bonds, and as you know, the hydrogen bonds are strong when there's a whole bunch of them together, but weak individually, which lets us break apart the double strands of DNA. Now, when we talk about them being bonded together, what we mean is their nitrogen bases bond in a particular manner, and we call those complementary pairs. Now, you can remember them with any kind of abbreviations or initials you like. You can think of a celebrity to remember that guanine has the complement of cytosine. So some people think George Clooney, so GC. So guanine, the complement is cytosine, meaning they can bind up beautifully. So if you have a double-stranded DNA, if one strand has guanine, then the second strand at that location will have cytosine because they're complements. If you have an RNA strand being made from a DNA template, if you have a guanine on the DNA, the RNA will have a cytosine because GC are complements. Adenine and thymine, so you can think of at someplace. So AT, adenine and thymine are complements, and that means that if you're dealing with RNA, the complement to adenine would be uracil instead, because you can, in RNA, replace thymine with uracil. Okay, if you have any questions or you're unclear what I mean by complements or anything on this slide, because I know the DNA, RNA stuff is sometimes a little confusing to students, just please feel free to send me a message and remind, and I will clarify. Now, just to help you visualize the difference, again, between DNA and RNA, there are quite a few differences, not just the thymine versus uracil, but the sugar that it's made up of as well. And again, the sugar is part of the nucleoside and nucleotide subunits. So remember, DNA has deoxyribose as its sugar, whereas RNA will have ribose. And you notice over here, deoxy means that it's missing an oxygen. So ribose has this oxygen over here in blue. Deoxy means that it has one less oxygen, but the rest of the structure looks the same. This slide also lets you visualize a little more about DNA. So one of the other big differences between DNA and RNA is that DNA you find in this double helix structure. And again, this slide is beautiful because it shows you the complementary pairs. So when you see GC, that's the complementary pair, and the complementary base pair of adenine is thymine. So for instance, this could easily be a multiple choice question where I say, which of the following is incorrectly matched with its base pair? Or I could say, which of the following is correctly matched with its base pair, its complementary base pair. So just always remember GC and AT. And again, anywhere that you see T, you can also substitute that for U if you're dealing with RNA. And then lastly, in terms of their structure, you have the RNA structure single-stranded, and again, not only is the single strand a difference, but the fact that it's uracil instead of thymine is another difference between DNA and RNA. This slide also lets you visualize the differences between DNA and RNA. So again, the sugar is different. The bases are different. Specifically, you can underline for yourself uracil versus thymine, double versus single strands, and the helix structure. And again, the reason why DNA has that helix structure, whereas RNA does not, is because DNA is double-stranded. And so that kind of interaction helps form a helix. And make sure you're comfortable with the differences between DNA versus RNA. So for instance, I can describe a strand and I could say that it has deoxyribose, thymine, and is double-stranded, and you should know that that's DNA. Okay, or I could say which of the following is a difference between DNA and RNA. Questions like that. Then we have the third type of nucleic acid, and it's another very important one because it is ATP, it's your energy, okay? So this nucleic acid, ATP, is made up of adenine, ribose, and 3 phosphate groups. Adenine, remember in the previous slides, is a nitrogen base. Ribose is a sugar, and then you have the 3 phosphate groups. And I want you to circle, star, highlight the 3 phosphate groups, because that is why ATP is high energy. Because by having 3 phosphate groups, when that molecule loses phosphate, when you have a break in the phosphate bonds, well, now you have high energy. It produces an unstable molecule with high energy. So in cells, what happens, one of the phosphate bonds gets hydrolyzed, and as you know, hydrolysis, what does that mean? Once again, picture that the fire truck hose spraying that high power water breaking bonds, right? So when you see one phosphate bond is hydrolyzed, it means that one of those 3 phosphate bonds is broken, and that releases high energy, okay? And so when we say ATP is an energy molecule, keep in mind is because cells can break that or hydrolyze that 3rd phosphate bond and release energy. Okay, and then the cell gets to use that. So here you have some figures to help you visualize what we were just discussing about ATP. Again, remind yourself that ATP is a nucleic acid. It has its sugar group here. It has adenine, which is a nitrogen base, and then it has these 3 phosphate groups. Notice that water can then break one of those phosphate bonds, and that releases energy that can be used by the cell. Okay, now that is the end of chapter 3, and again, I just want to give you a chance to see a little summary of everything. So that'll be our next slide, right? So here is the summary of all of the different biomolecules we discussed today. It reminds you of some of their subunits and examples, as well as the functions. But again, a slide like this, when you have a figure like this, and I didn't go through each of the individual parts, you would never have to memorize anything on here. And if it's something I did not mention in the earlier slides, such as here and cuticles and earwax, if I didn't mention it during the lesson, then you are not responsible for that on the exams, okay? So once you finish a chapter, always remind yourself what you need to do next. Make sure that you add Chapter 3 into your weekly study guide. Again, that could be in any format that you choose, but just make sure that you have all of the definitions, and make sure you have the questions that were asked throughout the lesson with answers. Okay, whether you include some pictures or whatnot is up to you. Keep in mind it will need to be in a Microsoft Word document that you'll then have to upload into Blackboard with scanning through Turnitin to make sure that there's no plagiarism of the internet or of your fellow classmates. Okay, you also have to always remember that by the end of the week, in addition to your study guide, you will need a discussion board post where you'll have to post your 5 questions, which you may want to include a question or 2 from this chapter. There'll be a multiple choice format, and you will also have to reply to a classmate's post as well. Any questions, anything you want more clarification of, or practice problems, or just to show me any of your work that you want to see if you're on your right track with regard to formatting or whatnot, never hesitate to send me a message in the Remind app. Like I always say, I have no life, so I do not mind helping you out. Right? Thank you and have a great day.